Welcome everyone to the last of our 2020 IMAG MSM working group meetings. I hope the individual subgroups will continue to meet, but this is our last plenary meeting, mini seminars and discussion. Today we have two mini seminars by Reed and Liliana, and then by Filippo. These are, of course, our short 15-minute teaser presentations, hoping to excite new collaborations and new ideas. I need to remind you that the meeting is being recorded. It's being streamed, live streamed, and it will be made available as a recorded document. So please bear that in mind. Myself, James Glazier, co-lead with Reinhard Labenbacher, web administration by Jim Sluka. You've all had coordination information by Bruce Shapiro. We should probably add Giuliano to the list because he's been handling Twitter and Slack issues. Remind you, we do have a Slack channel if people want to use it. It's available. We can also post information, and we encourage you to post information on the IMAG MSM wiki pages. So please contact us if we need any help with either of those communication methodologies. Then without more ado, we will start with a brief presentation and discussion by Reed and Liliana from NIAID, and I will turn it over to them.
Great, thank you. So Liliana is on the call as well. My name is Reed Shabman. Can you all see my screen? Okay, great. So I'm going to try to stick to time, and I'll be giving the overview. Liliana and I put together a few slides, and she may jump in. But I wanted to say thanks again, and just I'm appreciative of the invitation to present. And today I want to talk about data dissemination and modeling infectious diseases from a NIAID perspective. So both Liliana and I are in the Office of Genomics and Advanced Technologies. So the quick presentation overview, just my personal opinion and our office opinion on goals and challenges of multi-scale modeling. You all are the experts, and so this is from a program officer standpoint. I then want to highlight one program, the Systems Biology program, where this is a consortium that does some modeling activities, and highlight these just to make folks aware of individuals in the field that are doing some of this work. Give a quick perspective on data generation and deposition, and then jump into some discussion if anyone's interested. And unfortunately, I don't have a ton of time to go too deep into each of these, but just try to touch the surface. So we kind of see the goal. So when we think about multi-scale modeling of infectious diseases, I've tried to highlight here goals and challenges. And as you can see, the keywords are kind of the same. So that, in our mind, goals are to integrate clinical metadata, large data sets, and cutting-edge technology in the context of infectious diseases, and apply these findings to predict disease outcome and inform treatment options. Ideally, we'd be able to validate and obtain mechanistic insight from this data integration, validation through laboratory studies, perhaps from clinical specimens, where you have most biologically relevant yet imperfect data sets, and then have the ability to refine these. And I think on the flip side of the coin, the challenges are really, there are challenges with data integration. How do you integrate different types of data? How do you validate and reproduce model results both within your own group, but maybe across groups and geographically spaced? And then finally, how do you refine models to actually improve accuracy? So can we predict survival, or can we predict binding interactions with greater accuracy or greater forecast? So this is a slide that I borrowed from Michael Yeaman at UCLA, who's part of the Systems Biology Centers that I will highlight it here in a second. And this is really just to highlight the fact that we think of these different scales of disease of modeling. So you can model all the way from in silico to in vitro, all the way up to clinical models. And really, each one of these has a place. Each one of these is important, and each one of them has their own challenges and ways to go. So it's something that we think about often. And just drill down into specifically where are we sit in this, or who are some of the individuals that our office supports in this effort, is the Systems Biology Consortium for Infectious Diseases. And so this is what I'll be talking about for the meat of the talk. This is a community that integrates experimental biology, computational tools, and modeling across time and space to develop strategies to predict and alleviate disease severity across multiple pathogens. So there's 11 centers in total. They will focus on antimicrobial resistance, tuberculosis, influenza, as well as emerging infectious diseases. And the point of this talk is to highlight that they put out individual papers around these infectious diseases, but that we also have a working group, the modeling working group within the centers, that try to share ideas and efforts in those areas. My slides are not advancing. Okay, there we go. So the overall accomplishments of this modeling working group, and I can provide these links later if you're interested, but there's a total, it was formed in 2016. There's a total of 14 presentations on different modeling topics, each with slides and tutorials and code available. So these aren't really like seminars like I'm giving now. They're more the nuts and bolts of Jupyter Notebooks and how you actually might do a beast analysis. Workshops have been done before by John Monk and Bernard Paulson, where they looked at genome scale modeling at our annual meeting, and the slides are available online. And a number of collaborations that come out that have fostered publications. So this is a productive group. So, you know, UCSD and Baylor have joined teams, Sloan Kettering and UCSD, for example. So it's brought together individuals from across the different modeling spaces. And really, I mean, I think this group doesn't need to see this slide, but we do want to address complex questions through data integration and modeling. So we're interested in all different types of data: clinical data, genomics, transcriptomics, microbiome, proteomics, metabolomics, lipidomics, like glycomics. How do we integrate all this data to make outputs where we can look at pathogen host networks, predict patient severity, develop software, harness patients' gut microbiome to cure infection, and understand that a little bit better, and then to maybe advance drug treatments. And we're also very bullish about putting data into repositories. So I've highlighted just a small number here. These are NCBI, dbGaP, GEO, for example, and then also bioinformatics resource centers, which are also managed in the office where Liliana is the director, which is the BVVRC and VUPath DB. So I'll just give a couple examples of different models that have been, different outputs that have come from this program. And if you're interested in looking at these papers in more detail and reaching out to the authors, that'd be fantastic. So maybe some in vitro and silico approaches here are some examples of how you would model complex data sets. The Boston College group has looked, Tim Van Option has looked at evolutionary pathways to AMR in test tubes and both in planktonic and biofilm conditions. So here, not viruses, but are able to predict antibiotic evolution in vitro. Bernard Paulson's group has done a lot of work, and I just highlighted one picture here, which I can't fully describe, but they are really looking at how do you layer different types of data, right? So how do you take genome, GWAS data and metabolomic data and layer it on to start looking at AMR phenotypes, where you can look at resistant strains in red versus susceptible strains in blue when you're looking at both metabolic states and genome-wide associations of SNPs, for example. So it's ongoing work as well. And you can, on another scale, you could look at models of disease severity and survival. There's a number of examples. Here's two. One is from the Sloan Kettering group. Joao has shown that microbiota can be a predictor of mortality in allogeneic hematopoietic stem cell transplantation. So they essentially are showing that higher diversity of microbiota post stem cell transplantation in cancer patients results in a longer survival, or a higher level of survival in patients compared to a lower diversity of the microbiome. And they're able to develop models that are going to predict survival in these categories. You can also look at this in the context of Staph aureus, so mortality risk, profiling of Staph aureus bacteremia by multi-omic serum analysis. So they're looking at serum and taking a number of large-scale data sets to predict early and predictive and pathogenic signatures. And then finally, I think important for this group are pandemic response efforts. So can you model PPF, protein-protein interactions. Nevin Croman's group has looked at this with built networks of SARS proteins and then done molecular docking of antivirals, for example, and they visualize these in index, so you can look at these protein networks. And also, Doug Laufenberger's group has performed system serology, and Gela Doctor's group has done system serology to look at distinct serology signatures that are associated with prediction of individuals that will recover versus individuals that might succumb to COVID. And so that was a kind of a whirlwind, but just to point out that these are the types of data that are being generated and the types of outputs that are being generated from the systems biology centers. And I think one critical question that we think about every day, if not every hour, is how do you get data out into the public space so that it can be used for further analyses, either primary or secondary analyses. And so we made a flyer, or Grace Teets, who I think is on the call here, and Liliana made a flyer a while back, and it's called 3 Easy Steps to Share COVID-19 Data. And I put ... or any other infectious disease data sets. And it's very simple. It's to identify a specialized or general repository, deposit your data set, and this can include process data, and then make sure you add the appropriate metadata, because that's very powerful. There are a variety of different repositories that could be used. And again, COVID-19 data, this could be interchanged with any data, right? But there are specific COVID-19 hubs or SARS-2 hubs within a lot of repositories if you're interested in specifically that data. And so this would include things for reagents, BEI, structure, omics, immunology, assays. And if you go to the website, you can get a whole list of some of these repositories. And then you can also deposit into generalist repositories. So on the previous slide, these were specific repositories. Generalist repositories such as Dataverse, NIH Figshare, these are all places that you can deposit data in any format that you like. And I think that doesn't solve the problem, but I think it's important to get data out there. And then there's a challenge of identifying metadata and data to leverage for multi-scale modeling. The Systems Biology Consortium is also trying to aggregate data. So outbreak.info is a great aggregator of disparate data into a single website. And so you can start to look at different types of SARS-CoV data, and maybe you can find information there that might help with some of the analyses that you're doing. And this is an ongoing effort, as well as generating sort of a searchable digital collection of systems biology assets so that others in the community could identify those. And so finally, I highlighted one program. There are a number of programs, other programs that are data generators that one could leverage for multi-scale modeling. This involve CREED, SEERS, and CIVICS. These are viral programs. SEERS and CIVICS have an influenza focus. CREED is emerging infectious diseases. There's an RFA Adam pointed to here that's undergone review, and this is for the antibiotic-resistant bacteria. Clinical units such as the CIVICS again, and the VTEU generates data, and these centers can develop and improve animal models, develop reagents. They're establishing longitudinal cohorts, and they're expanding human challenge study capability and capacity. And so I would say thank you, and take any comments or questions. That was kind of a high-level overview of our perspective of things. But if anybody wants to talk more about data, deposition modeling, specific modeling efforts, or future collaborations, you have to.
Okay, thank you very much for the overview. That was impressive, comprehensive in a short amount of time. The slides and information will be available for people to refer back to. So we have time for a few questions.
This is John Rice. And how much involvement do you have from the zoonotic science side? You have the veterinarians and the animal scientists who are actively getting interested in what's coming out of the human research that they could use?
It's a great question. Liliana, do you want to have any thoughts?
Well, I would say, for example, when we had cases where there's zoonotic transmission, we have had people in that space. So the group in Tulane that collaborate with the Systems Biology Center for Emerging Viruses at Scripps, they have a group of veterinarians and other people who would be more studying environmental transmission, or not environmental, but zoonotic transmission into humans. And then through our Sears networks that Reed talked about for influenza, we have a large surveillance effort in waterfowl and other types of environments where you could have the admixture of the viruses that would then jump over to humans. So I think there is involvement from the types of people who would be generating this type of data.
But it's not as strong. It's an area that we might explore improving. True?
Yeah, I think in general, not only zoonotic transmissions, environmental exposures as well, and how do we detect things in the environment, and whether there's any thresholds that would initiate some sort of alert or something for infectious disease surveillance. Yeah.
Thank you.
Reed, this is Reinhard. Can you maybe say just a few words about the grant program that supports these centers?
Sure. These are, let me just step back. Yeah, so just to add on to Liliana's comment, Bob Gary is in that systems biology grant that Liliana was referring to, and they're looking at rodents and the reservoir hosts for Lassa, and so in West Africa. And so that's an interesting, but I think there is more to be done there. The systems biology program was established in, around 2008. I'm gonna let Liliana correct me in a second. But there's been a number of iterations. These are they started as contracts, they're now grants. They're UO1, U19 programs in 5-year intervals. And currently the latest iteration of that U19 is around, it's about halfway through. And so, the office that I'm in now has recognized these holistic approaches with large data and modeling as an important attribute and a gap that needs to be filled from a consortium level, because we think that folks are from different backgrounds mixing back are better together than apart. And so that's kind of a high level.
Good, thank you.
I'm looking to see if there are any other questions. I'm always intrigued personally because one of the things I have as a sort of a slogan is that ultimately properly annotated data should be able to be turned into executable models, and models themselves, if they're annotated properly, should be able to embody knowledge and exist as data. We're a long way from being able to do that, but I have a personal belief that we need to try to bridge the gap which we see between models and data, and to try to merge those into something that's more unified. It's not an easy problem to deal with, but you maybe have thought about that.
We have, and I think our groups have talked about it quite a bit, both internally and extramurally. So I'd say one of the concepts and ideas that we're trying to really push is to get people to deposit not only the raw data from their experiments, but also the process data after they have done their cleanups and everything else, right? Because so much of the downstream effort really depends on how you do that first step. And many times that process data, then analyzed data is only available in a table in a paper. And so you don't really have that in a computable format. So we're trying to really address that and making sure, and we post there in our handout said, not only just your raw data, but also your process data. So that's something that we're trying to convey to the data generators, that is imperative and important, and also to make their workflows and their protocols of how they actually process the data also available and ideally reproducible, right? So we had a really interesting conversation with the editors of eLife, who are really trying to make sure their articles are fully reproducible, right? And so that was, we were already talking about that, and then we had them give us a talk at the, I think it was, I don't remember which one of our working groups, but really that's the objective, because I think data on its own is good. Data with all of the other elements and process data is even better. And similarly with the models, we've had a lot of conversations about what's the best place to deposit the models, how do you deposit, how how much annotation and do you put into the models themselves? And so these are all ongoing conversations that I'm sure you're having here, and we're having similar conversations in our working groups.
I'd love to talk more about that at some point if you like. So, and I know there are many people in this community that are interested in aspects of this. So that's great. Is there time for one last question before we move on? There's so many things that were raised as interesting.
Yeah, if I can just ask a question. So my name's Mitchell. I'm a graduate student at North Carolina State University. I just want to ask, from your perspective, do you approach the machine learning kind of strictly data-driven approach in a different manner than mechanistic modeling when you're looking at funding and grants and what you're trying to get done with your organization?
So do you want me to take that, Liliana, or should
Go ahead, Reed.
all right, I'll say, so the short answer is we're interested in all different types of models, whether that's pure computational or if there's a lab component or a mechanistic component. The bottom line is there's also an Office of Data Science that is pushing a lot of things, and there's actually a council approved concept out there that's specifically highlighting just novel approaches for computational modeling. So we can drop that into the link. It's not a foe on the street, but it's a publicly available concept that's been approved by NIAID Council. Now, but going back to your original question real quickly, Bernard Paulson is kind of a leader in our systems biology with a lot of this type of computational modeling from just data, secondary data analysis, and we think is an important component.
Great, thank you.
And Reed, a quick request. Our working group has a subgroup for dissemination, education, training specifically as a subgroup. And follow up to this, I'd be really interested in hearing some suggested names of people who know what you're doing, who might have a little bit of time to be part of another working group and help us not be reinventing wheels. And so if you have a candidate in mind, please share. Thank you.
Okay, thank you very much. I'm sorry we have to move on. Thank you again. I hope we can have you back and talk more in the future. Really appreciate your participation. We're going to move on now to Filippo Castiglione, who's staying up very late to speak to us from Rome. And he's going to be talking about the SARS-CoV-2 infection and cohort study in silico. And I'll turn it over to Filippo.
Thank you, James. Let me try to share the screen. It should be this one. Do you see my PowerPoint?
That looks fine, thank you.
Okay, so let's put it widescreen. Okay, so 15 minutes, I want to tell you a study I have done with the computational model, which is multi-scale, which I'm not going to describe in detail because there's no time. The question I wanted to address with the model was how can we explain the high variability of a clinical presentation of the COVID, basically, because people know that, I mean, from the very beginning, it was clear that the mortality rates was quite diverse in different countries. And there was a very difference in clinical presentations. People get mild disease, others die, and there's no correlation direct apart with the age of the individual. So the question was, can we try to recall this diversity just by focusing on two characteristics of the infection? And one is the host, let's say the health of the host or the immune competence of the host, and the other one is the characteristic of the pathogen itself. So two different competitors and generating such a huge diversity. So basically we use this computational model, I will tell you in a second, to try to test this idea. The model is an agent-based stochastic multi-clonal model with the different cells types, molecular types, interleukins, and so on. And with the many immunological, let's say, hypotheses or processes or whatever functions in it. So it's quite detailed in terms of the biology or the immunology, which is described in the model. So the two components of this study were the level of immunological competence, which we simply represented with a negative correlation with age. So we have this parameters IC, immunological competence, which decreases with age. Okay, it has been a little bit arbitrary, the slope of this curve, but somehow we the aim was to try to reproduce the final distribution of causalties in the age classes, okay, which is not linear. This is important. So this the host factor basically derived two characteristics of the immune response. So that parameter IC was used to determine two other parameters connected to the ability of antigen presenting cell to phagocytize the virus and present. So it's somehow related to the speed of the innate immunity to kick in and start the adaptive immunity. And the other parameter that we somehow tuned with this immunological competence parameter, let me put the laser, was basically the leukocyte count, which in the model is usually represented as a statistical instantiation of this distribution, which are normal range reference in humans, that we basically reduced, like simulating a kind of a lymphopenia due to age. It is well known that there is a relationship between age and the ability of the immune response to protect. It's called, in fact, immunological aging. So this concept is coded in this relationship. On the side of the pathogen, we coded two parameters. One is the viral load, the infective viral load. So basically the amount of viral particles at time zero of the simulation. And the other one is the probability for the virus, one viral particle to enter into a target cell. Basically the affinity to the ACE2 molecule from the spike molecule of the virus. So these parameters basically identify one simulated virtual individual, and we reproduced 1,500 simulation per age class. So we're talking about a large number of simulations, basically consisting in different trajectories in this stochastic simulation. And then we analyzed the result, and we had to match reality. We had to somehow find a threshold theta, with which we could classify outcomes of the simulation in three classes. Those belonging to the critical classes are virtual individual which at day 30 of the simulation have still a high level of viral load. So people who have not, virtual people who have not controlled the virus. Then we have the partially recovered class, which is people who have reduced considerably the viral load, but not cleared completely, while the fully recovered people or individuals are those who have cleared the virus before day 30 of the simulation. So we were observing only the first 30 days, so the initial phase of the acute phase of the infection. Of course, so this is the picture looking at the viremia. We recovered these three different behaviors, and to note this, the partially recovered class includes the asymptomatic, because those people have a very low viral load, but they are not cleared. So somehow the immune system is coping with the virus, but not completely. And on the side of looking antibody, the same equivalent simulation look like this one. So it's interesting the fact that critical individuals have a very variable level of antibody titers, and which somehow include the class of late responders, people who have, you know, a late, let's say, delayed response to the virus, or people who have a very weak response for different reasons, of course, related to the age and indirectly to the leukocyte counts or immunocompetence, let's say, which is coded in the model. So we we tune the parameters theta and other parameters I've just mentioned by trying to reproduce the death rate by age at a certain point in history of this pandemic. It was February, I think, or March, something like this. And starting from this, we try to extrapolate and see other statistical outcomes of this virtual cohort. Okay, so we reproduce reasonable parameters. I mean, these numbers here have not been tuned directly. So the fact that in vivo the virus is visible in the pharyngea after 4 days is somehow close to the fact that we have a peak in viral load between 3 and 5 days in the simulation, and so on and so forth. I'm not going into the details, but some of these are very well in agreement to what is observed or at least it was observed in March, April. So the remaining observation are the outcome. And so this is in agreement also with reality, the fact that the viral load correlates with disease progression. This is not a novelty. More virus you get or you are infected day 0, and of course worst will be the final outcome. This is clear for, I guess, any pathogen. The other fact is that IL-6 correlates with disease severity. So the people who have go critical basically show an overall higher level of inflammatory cytokines, and that this is more or less, let's say, constant by age class. So it doesn't discriminate by age class. What discriminates is really different by age class is the level, the type of immune response elicited. Cytotoxic response, for example, it is smaller for aged people, for elder people, than compared to younger people. And interestingly, critical individuals have a stronger cytotoxic response than people who recover. And the reason is it seems a cause, but it's a consequence of this fact, the fact that the humoral response is more important in controlling the virus. At least this is what the simulation says. And as you see, those who are experience a critical outcome have a very poor antibody response. And so perhaps the fact that the cytotoxic response is higher is just a consequence. It's the immune system trying to cope with the infection. Many more cells are infected, more possibility for cytotoxic cells to expand. The final result we wanted to see if we could, by looking at the number of cytotoxic cells at day 30 and the antibody titers, if we could somehow predict the outcome of the virtual individual and the class, so if critical or not critical. So we use a logistic regression to split apart these two sets of recovered and critical with its typical machine learning procedure to split these two things. And we found a nice precision and recall score, but only if the antibodies and the CTL, that means the photo taken to the virtual individual, is done after day 25. So before that, the information itself is not sufficient to predict the outcome, the final outcome at day 30. And this is basically what I wanted to say, and I will be happy to take questions if you have. I hope I stayed within the 15 minutes, maybe even shorter. I don't know.
Thank you very much. That was a real taste. People will be hungry for more information, I'm sure. Please type for a question or two.
I have a question if you can hear me.
Sure. Yes.
So I noticed that you model both mortality and the viral load. Do you have, and this is something that I currently am looking for, do you have a profile of when a person dies if they die? Meaning how many days from the beginning? Do you have models for that I can use?
Well, it's not the model, it's an emerging property. The definition of what is dead in my virtual individual is somehow artificial, because of course this is just a model of the immune response. I count cells and cytokines, so I have to set a threshold somewhere and say, and set apart what it's, let's say, in trouble with respect to who is not in trouble. I didn't show it in the slides, but one thing I did was to estimate the damage, let's say, in terms of how many epithelial cells are killed by either by the virus or by the cytotoxic cells. And that is that correlates quite nicely with the mortality, with what I believe is a severe case.
Okay. Do you have several assumptions that can be tested?
Of course, yes. The model can be used in many different ways. Of course, the next step will be to put it side by side with some clinical, not statistical. But the problem is that when you go and look at the specific patient data, it's very difficult to have a very nice fit.
It's all understood. However, if you have a functional form or even a numerical form that says this is what the chances of a person dying in day 1, 2, 3, 4, up until like 20, I can use this. And if it's only an assumption, it's also good. If you have several of those, it's also good.
So could you, Filippo, maybe you could you share a link to the paper or the preprint describing the model structure?
Yes, please.
Yeah, I will. Well, we still have to submit the paper. I didn't put it in the preprint. I could do it. It will be soon there, probably in a week or something. So
Okay.
I think
I will bother you.
Sure.
I think we'd all love to see that.
That
would be great.
Wonderful.
Time for one more question. I hope we can certainly continue conversation offline. Other question? I certainly would like to. I was just love to see the model structure and organization. So that'll be. Look forward to that.
Thank you.
I know it's late for you, so appreciate your taking.
Thank you. Thank you.
Other questions? No? Okay.
I have one quick question.
Yeah. Gary, go ahead, please.
So this is great work, and I've followed it for multiple years. So fantastic. So have you started trying to simulate hypothetical interventions on this, like interdiction of the various pathways or cytokines that you might have, right, to like test, for instance, monoclonal antibody therapy against inflammatory cytokines and stuff like that?
No, I have done, but it's a very nice. It would be a very nice way to use the model, actually.
All right. Thank you.
Yeah, absolutely. Yeah. Thank you.
Lovely. Thank you again. So
I have to stop the share. Yeah.
So thank you again, Filippo. That was very, very intriguing and again, very relevant to both talks. Thank you to all of our speakers for today. Reminder that this is our last meeting of the year. No meeting on December 24th. I don't think, even with the world which has canceled Christmas, I don't think people really want to have a seminar on the Christmas Eve, probably New Year's Eve as well. People will be too eager to have the year over to want to have a seminar. We'll begin again January 7th with Paul Macklin and Bart Ermentrout speaking. And then Grace has agreed to speak on the 14th. And we'll have a talk also from Purdue. We're very eager to have people speak. Please contact us if you'd like to speak. You don't have to be a working group member to speak. So if you have suggestions for speakers you'd like to hear, please send those to us and we'll do our best to accommodate those requests. It's been a great seminar series so far. I hope it's been useful for people. Now I'll turn it over to Reinhard, and we're going to, for those subgroup leaders who are present, we'd like to quickly review the status of the subgroups and have quick reports. So Reinhard, over to you.
Yeah, thank you, James. First of all, let me also thank the presenters. They're extremely interesting and stimulating. So as everybody knows, of course, the bulk of the work is being done by the subgroups, and we have a significant number of these. What James and I thought would be useful is to just have a brief update on what all the subgroups have been doing. So if those subgroup leaders who are here, we could each of them have present a 1 minute, really a 1 minute update. We have 15 minutes left. We have 30 subgroups. So hopefully not all 30 subgroup leaders are here. If not, we're going to have to stay over time. But so let's get started. And maybe the easiest, so simply say, whether you've met, what you've done, what you want to do. And maybe the easiest would be if we just go to the list of subgroups and work our way down. That might be the most systematic way. So I don't know, James or Jim, whoever is controlling the slides. Here we are. So let me just we can as we go down, maybe people can just speak up. So let's start with physiological models, innate and adaptive immune response.
Yes. So last week, this is Ruchira, one of the subgroup leads. Last week, the three of us leads met directly after this meeting, and Alyssa Schwartz hadn't been able to meet yet. And what we mostly discussed were how to organize the subgroup. So there's two main things that in this goal of surveying the literature, how would we divide the labor up among the subgroup members? Would we be able to make a roadmap before having a meeting? And the other is how would we interact with the other groups? So focusing on the first thing, we sent a Slack message to the working group organizers asking, can we have access to the forms that people filled out when they submitted their interests? Because they submitted quite a bit of interest data, and we haven't gotten a response to that Slack message, but barring that, we could ourselves again ask for people's interests and see how we can organize the group around that. So that's what we've done.
Great. Thanks, Ruchira. Host pathogen interactions.
Yeah, this is Yaling Lu here. So we haven't going on too much yet. We're going to fill up the needs. It's going to meet tomorrow, Julie, Winston, and me, to discuss how we're going to organize this group. So it's kind of slow, but we're trying to pick it up.
Okay, great. Thank you. Tissue damage and recovery.
Hi, this is Yi. This is Yi Jian. I met with Kai Ming Ye, the other lead. We discussed potential mutual interest amongst ourselves, and I set up a doodle to ask for other interested people to potentially to meet this week, but I haven't gotten enough feedback. Maybe 3 people sent me available time so far. So that's probably not going to happen this week. So we'll find out a different time.
Okay, great, thank you. Let's move on to virus models, viral transport. Okay, how about viral replication?
I think there are two of us here. This is Jim Fader. We haven't met yet. I set up a Slack channel for the group and put up some comments and solicited some feedback. It's been pretty light so far. I suppose we should come up with a plan to meet. Things have been wrapping up for me for the end of the term, so I've been a little bit busy. I don't know if Unyang would like to, I hope I got her name right. We haven't met in person, so or even virtually as another lead on this.
Oh yeah, I got your Slack channel open up and I saw all the emails. I've been pretty busy with other facts, so I didn't have a chance to really visit, but we can set up the meeting soon.
Yeah.
Thanks.
Okay, great. Thanks to both of you. Viral evolution.
Hi, this is Fred Adler. Yeah, I'd like to second what the previous group said, which is haven't gotten very far yet, but I can beat them in that I had my other co-lead quit and decided to be demoted. So the leadership has met in the presence of myself. I've reached out to the group and heard back from a few people, but I've sort of laid out an outline of what we would do, but was not going to try to attempt to meet until early next year, given everyone's constraints.
Okay, great, thank you. Therapeutics and medicine. So here we have drug development.
Yes, hi everyone. This is Robert Stratford, and Jim Sluka and I have met. Thanks very much to Jim for setting that up. And we have a regular standing meeting bi-weekly, and so we're moving forward. We've got Gary on, who's also joined us, and so we're thankful for that. So it's a fairly small group, but we're looking for new volunteers, and somehow we hope to reach out to people in the pharmaceutical companies. We think that with their wealth of experience and even ongoing work that they're doing in the antiviral area, maybe they could be very, very useful to this group.
Great, thank you, Robert. Vaccine development. Okay. Modeling individual responses to disease and treatment.
Hi, so Taryn and I had a phone or a Zoom call several weeks ago. I have a draft email soliciting information from the working group that'll go out after this call. And the hope is trying to collect some feedback on kind of a mission statement for the working group, establish kind of tiers of levels of interest and engagement, and then try and set up some sort of more interactive video conference sometime in January.
Great, thank you, Gary. Modeling decontamination of surfaces and materials. Machine learning for health monitoring.
Is Guanling's not here? This is Yijian again. I met with Guanling and discussed what we wanted to do. So we sort of agreed on eventually having a white paper, but first thing would be to see what kind of data is out there for machine learning in terms of COVID-related health monitoring or other diseases-related health monitoring. He sent out an email to the group, and one graduate student from Caltech responded. So we have been helping him with his machine learning project for the COVID aerosolized particle settling time and how to use that to predict COVID cases. So that's where we are for this group.
Okay, great, thank you. Moving on to individual organ systems, the lungs.
Lungs is me again. Again, a doodle without much response. So waiting for next week.
Okay, very good. You're really working all these things. And heart and vasculature, right? Kidney and liver, comorbidities.
It's me. That's me. It's me, and now I have a co-lead. We actually, there was an email sent to about all the group individually. About half responded, about 10 people out of 20. We found a co-lead. We're going to continue reorganizing, but it seems that it's a small but very active group. We'll see how it goes, but it seems promising.
Okay, great. Thank you, Jacob.
Oh, sorry. Thank you, Jacob. I took the lead of this group. I am Gilberto. Sorry, I took the lead of this group like 2-3 days ago.
I know.
so I was, finishing grading, and I hope that we're going to have a meeting in January. Okay.
That's fine. We'll talk over email.
Okay. Thank you.
Okay, terrific. Thanks very much. Data, experimental and clinical data. Santiago. AI-based data processing. Then we have so far an orphan group in infection in experimental models. Moving on to zoonotic reservoir animals.
This is Gary again. Taryn, John, and I have talked about this a little bit. This is a little bit more an evolution. There's been a lot of kind of outreach to different types of groups in terms of trying to get some animal biologists engaged in this. This is something I think that we are in the process of trying to formulate a strategy a little bit more. It was good to hear some of this interest from the NIAID systems biology groups. John and I had some pre this pandemic working group along with James regarding some bat biologists. And I think that there is a lot of initial outreach that's required because the concept of multi-scale modeling within that particular domain, at least amongst the people we spoke to, is just not as robust. So I think trying to formulate an introductory strategy is just a little bit trickier. So this is a little bit more in evolution.
Great, thank you, Gary. Emerging and re-emerging diseases. All right, then moving on to modeling technology, aerosol transport in lung, lymph, and blood.
Yes, so this is Chantal Darken, and so what we've done so far, it's really only discussing between the two leaders, me and Marin Tafa in New Zealand, and an email to the group should go out next week, and the plan is to request not only a bibliography like what's listed in what we would have to do with target dates that was sent a few weeks ago, but also to second, I think the first person that talked, ask also every member to send a short bio with their main focus in this multi-scale modeling so that we can use that to start getting a skeleton of what the white paper would look like and if we have holes in expertise. And the other thing we will ask our members is to help us recruit additional members to the group from their colleagues that they know working in similar field. So that's kind of where we are. So we haven't engaged with the members per se yet, but that's working, I mean, in progress.
Great, thank you, Chantal. Knowledge acquisition and modeling. And then there's a crowdsourcing software development, the name really should be crowdsourcing models. So that's the co-leads are me and Paul Macklin. So we met earlier this week. We had about eight people there, I believe, 8 or 9. And we had a spirited discussion about what the issues were. There really sort of emerged three different components that need to be addressed. One is, of course, platforms that allow crowdsourcing integration of models. Another one is models, how should they be developed? And the third one really is the social ecosystem. Let's call it using a modern name that allows people to to work together in appropriate ways. We're going to have bi-weekly meetings. We will put together a white paper that frames this issue that might turn into either a review or a perspective paper, and that's where we are. Then there's model standards. Jacob.
Yeah. So it's actually standards is kind of a problem if you actually say, because there aren't real standards, not the ones that SDO, at least not that I'm aware of, not the one the community really uses. There are standardization efforts. So in short, Rahman Sharif joined us as a lead, and they've been doing a lot of work on looking at all sorts of models and figuring out how reproducible they are. So we probably will change the name of the group, and I say probably because it's still, we have to address the group, but it will be a model reproducibility, credibility, and standardization rather than standards. Standards will be misleading people who think that's standards. So this is currently where it stands. We're going to launch a web page and a mailing list probably over the weekend, and then we're going to address the group. It's a big group. So far, it's not very responsive compared to the smaller groups, but we'll keep going and try maybe targeting individual people, see their work on any of those issues. If anyone has to do anything with annotations, we want to know, because we're probably going to drop it from the name. So if anyone wants to say something, holler. That's it.
Okay, great. Thank you, Jacob. Moving on to social issues, dissemination, training, outreach to the public.
Yeah, this is John, and we've been fairly busy. We have help in the leadership side, and Jim Sluka has joined us as one of the co-formers, and he's our liaison to the parent group. One of the questions is, how do we refer to the main body of the working group when we're all subgroups? So I call you the parent group for now. We had 36 people had said they were interested in the dissemination related topic, have gone out to them several times, kind of followed the lead of the big group by having a form for them to fill out if they wanted to confirm their participation. We've got 15 people who have replied positively, and that included asking them if they could kind of consider maybe 6 hours a month commitment over the next year, hour and a half a week, and beautifully most have said that was a reasonable request. So we think that the 16 that we have are going to be active members and not just observers. Looking at the scope of the group, our question is, what is the scope of the group? And we talk about, to whom do we disseminate? Easiest way is that the range of dissemination is within the parent working group, which I say is talking to ourselves, but it may not be as easy to talk to ourselves as we think it is. And the other extreme being the general public. In the responses of people who want to be members of the group, there was a surprising number spontaneously that brought up the issue of the loss of some credibility of science and modeling because of public experience in the last year. So we anticipate that's something we're going to have to deal with. And overall, the model for our landscape is one of who, what, and how, like identifying the range of audiences for dissemination, education, and training, identifying the kinds of information that we could imagine having to get out, and then talking about the methods. And maybe this is where some of the research parts will come in the methods for delivering it. And also a heavy interest in STEM, which I'm extending from STEM K to 12. I now write STEM K to MD, because well beyond 12th grade, we have a lot of people that need to get retrained and re-educated.
Great, thank you, John. James, could you maybe take over for the last two, because I have another meeting that I'm late to that I need to take. So thank you, everybody. Sounds like everybody's doing things.
Okay.
So have a great rest of the year, and we'll get together again next year.
Okay, I guess is Bobby on the call to discuss health equity?
Yeah, I'm going to Melissa's for a while. You got.
Okay. I think actually we made it through our list pretty much. So I'd like that. Yes.
You have the merged groups.
Yep. Yes, that's fine.
So we merged two integration related working groups. There was a lot of overlap of people. Both groups that dealt with integration is now called integration work subgroup. We have launched a SimTK site, and we're going to start inviting people to the mailing list. All of those who actually signed up, we expect them to start appearing in the mailing list and start a discussion at some point, but we're going to push more things. Yaling is also here, or was at least, so he's one of the colleagues. We have like 3 colleagues because it's a big group. Robin as well, and Ruth, but that's it for this integration group.
I want to apologize for everybody if I haven't responded to email. I've been trying to focus on keeping the overall thing going, which means I haven't had as much time to work with the individual subgroups as I'd like. I want to thank everybody for participating. Thank you again, Filippo, Reed, and everyone for speaking today, for your contributions. Thank you for the subgroup leaders for actually getting things started. That's wonderful. Look forward to talking to you on and offline and seeing you in the new year. I think it's going to be an exciting adventure to try to see how this group evolves. And so best wishes for the holidays and the new year. And we will see you if there aren't subgroup meetings and subgroup meetings before then, otherwise on January 7th.
Thanks to you.